My name is John McCone. I'm from Merrimack River Watershed Council. We are a nonprofit. We've been around for about 50 years, and our mission is sort of simple but very complex. The simple part is to make the river cleaner and healthier for the next generation. The complex part is actually trying to do that, because the river is constantly changing. We find new challenges. Most recently, climate change, I think, has been on everyone's mind, and we see a lot of influence of that on the Merrimack River. But we also see a lot of successes, and Micah is going to talk about one of them tonight, which is the rise of the sturgeon population. Has anyone seen a sturgeon in the river? Sure. Isn't it spectacular? It's really something. If you haven't, you got to see it. It's really quite a sight. So we do a number of different programs. If you're interested in getting involved with the river, we do cleanups, we do a lot of water testing, we do different sorts of projects up and down, like education projects. We also have a project right here in Newburyport, just outside our window. You can almost see it. We brought in a very high-tech bacteria sensor. It's the same sort of sensor that was used by the Olympic Committee in Paris to study the Seine River and see if it was safe for swimmers. So we have that exact same sensor right here in Newburyport now, and it's telling us a lot of information about bacteria levels and something called combined sewer overflows, which happens in this river, also happen in the Seine. Yes?
What is again the Merrimack River?
Watershed Council. It's a mouthful. Yeah. So we got to, that's one of our missions, is to think about changing our name. So I better tell my boss that. So anyways, so that's what we do. You know, we work with a lot of different people, and there are many people who work very hard to clean up the Merrimack. It is a much cleaner river than it was 50 years ago. One of the people who is absolutely on the top shelf of champions for our river is Micah. Micah Kieffer from the United States Geological Survey. Is that right? Yep. And so Micah has dedicated his career to this river and to other rivers in New England to study the movements and the ecology of sturgeon that live in these rivers, and to basically try to make them healthier, you know, and to increase their population, for us to understand them, their behaviors, and how we can help them best. Micah does a lot of different jobs. You wear a lot of hats at USGS, I see. You're in charge of their fleet of boats. You're a diver.
Collateral duties.
Collateral duties, right. And, you know, he also works on other rivers as well in New England. His work has been instrumental in setting policies and decision-making for sturgeons around. So Micah has done just a fantastic job for this species. He's a real champion for it. Micah, thanks so much for being here. Are you ready?
I am ready.
All right.
So I got a computer, I got a laser pointer, and a microphone. Well, let's see what happens. Thank you for that warm welcome, both John and James. It is truly my honor to be here, and to see this kind of interest in sturgeons is inspirational to me, because you know I work for the federal government, so I'm working for you guys. And whenever I encounter people in the wild, I take opportunity to try. If I happen to have a sturgeon on my boat, I'll step out and try to show people these incredible animals. And I always end the conversation with, you know, they're your fish too. You know, we're all stewards of the natural resources that, you know, remain. And so part of my job is to, in many ways, I have a really, forget my hand because I do talk with them. My job is to go out and learn about the animal. So, you know, I get permits, I have to find funding, and come up with study plans that are of interest and value primarily to the people whose job it is to manage the sturgeon. Now that's not my job. I'm not involved in any policy decisions or the enforcement of laws. I go out and be one with the sturgeon and try to learn what's important to know about protecting the animal. And if you're aware at all about conservation of animals, you may have heard that protecting their habitat is as important as protecting the fish themselves. So part of learning about an animal is to what are its peculiarities, its preference, etc., and what habitats does it rely upon. So I'm gonna first start off by talking to you, give you a little bit of background about what is a sturgeon, what is its life like, what are its unique features and peculiarities. Then I'm going to talk a little bit about how I go about doing my job. How do you learn about sturgeons? What does that involve? And that's the fun part for me anyway. Then I will talk a little bit more about some of the really interesting and unique things we've learned over the past decades that I've been out here chasing these fish around. I'm sorry, I can't remember what comes after that, but I try to minimize the data-heavy slides. It's more like a picture book on a coffee table, the photographs that I'm going to be showing you. But there are some data slides, and I'll nurse you through those to make some of the points that I think are interesting. So now I got to find the right button. So sturgeon life history. Someone just said tonight, the sturgeon flopped in my boat, and somebody said it looks like a shark. And it looks very much like a shark. In fact, its body is designed just like a shark's. And what can sharks do with that body? They can migrate long distances. And so can sturgeons. So it's a very good design for running up rivers, hundreds, sometimes thousands of kilometers. What they can't do very well with that body is make tight turns in a confined space. A salmon or a shad can enter a fish ladder and negotiate those turbulent tight turns. Sturgeons can't do that. They don't bend as much. They're not as quick to make those kind of movements. So, and I'll get into that a little bit more later. And you see there, they've got there's 5 rows. There's 2 on the bottom, 1 on each side, and 1 across the top of these kind of bony plates. And I have one right here. And this is called a sturgeon scute. And that's the technical term for it. But it is, when you think of a fish and scales, this is kind of the ancestor of a fish scale. So as fish evolved over time, this armor changed into something that's a little bit less cumbersome and gives them more freedom of movement. But this is a fish called placoderm, which means skin plate, and this even has more armor. And I'll get to why that is in just a bit. One of the most interesting features of the sturgeon is this mouth. It's called a protrusible mouth, and it's unique amongst the fishes. And it actually comes out like a tube, and it's got musculature and bones and cartilage to allow it to spit water into the substrate and stir it up and eat things that are living right at the base of the substrate. So I knew people that did their PhD work on figuring out how that thing evolved. So we call sturgeons a living fossil, and that means simply that the basic body shape of the animal is the same as what's represented in the fossil record. So it depends on who you ask. They think that sturgeons were around for about 200 million years. And there's no sturgeon in this picture. I put this in there because it grabs the kids' attention. But this is kind of sturgeon-like, but these kind of pelagic predators that lived years ago, I mean, they ate turtles. So this bony armor that sturgeons grew has a very, very prominent place 200 million years ago. And what's unique about sturgeons is that their body design and behavior was so successful that they didn't have to change for 200 million years. Now, if they could evolve a pair of legs and to climb ladders and get up over dams, they'd be golden. Right now, that's kind of one of the greatest impediments for sturgeon recovery. These two images are actually chunks of fossilized sturgeons, and they were taken from a deposit that scientists believe occurred during the dinosaur-killing asteroid that slammed into the Yucatan. And so there was hundreds of fish fossils. So, you know, when you have big tidal waves that come through, a lot of animals die, and a lot of times they get pushed into the same kind of a collection area, and then they get fossilized. So there's lots of different stuff in there. So there were paddlefish and sturgeons, and it was dated to be about 65 million years ago when that asteroid was theorized to have changed the course of dinosaurs. So while the dinosaurs were gasping out their last, the sturgeons survived, and in fact they have survived 2 mass extinction events on the planet. And ocean animals have some level of protection from terrestrial calamities. So I don't know if you've ever heard of the fish called the coelacanth. That was a fossilized, believed to be extinct fish until one was captured in 1933. Now there are actually scuba divers that have gone 600 meters off of Madagascar and took films of living coelacanths. So the global distribution for sturgeons is confined to the northern hemisphere. And I tried to make this isn't a precise band of where sturgeons live, but there's none in the southern hemisphere. But I took caution to slice Florida in half. And then there's a reason for that. There's 2 species of sturgeon in the Merrimack River, the shortnose sturgeon and the Atlantic sturgeon, and I'll get to that in a moment. But the Atlantic sturgeon, its range used to be all along our eastern seaboard and into the Gulf of Mexico. But that was before the state of Florida emerged as a peninsula. There was no Florida at that time. So there was a single species that was inhabiting that range of river, and then the Florida Peninsula arose, and all of a sudden now it made it too far south for sturgeon to continue to live or to move back and forth between these 2 bodies of water. So they diverged, and now there's an Atlantic sturgeon and a subspecies called the Gulf of Mexico sturgeon. And one has a smaller spleen than the other. And that's how you tell them apart. But you know, that's, you know, there are people who have devoted their careers to try and figure out which animal begat which animal and how did evolution work. I mean, Darwinism is kind of the same sort of principle. We see these animals that look very similar, but there's a slight difference in why did that happen. In North America, there are 9 species of sturgeon. The 2 that I'm going to talk about tonight that inhabit the Merrimack River are the Atlantic sturgeon, and here's the short-nosed sturgeon. And what this graph deceives you by is that the sizes of the fish, the different species relative to one another, is not accurate. They made them all the same size because it makes a nice poster. But the shovel nose is the smallest, inhabiting the Mississippi drainage. And an adult is about 1.5 feet, maybe 2 feet long. The white sturgeon inhabits the Columbia River Basin out west, and they can reach 20 feet long. So this is an order of magnitude difference in size. And there are even bigger sturgeons in some of the European rivers. So the most charismatic being the beluga sturgeon, which is critically endangered at this point in time. And they're working on the Danube River to try to restore And they've got 4 species of sturgeon. So, sturgeons are a migratory fish, and the term diadromy is a catch-all categorization for fish that have some form of migration in their life history. And there's 4 types of migration that I'll talk about. Anadromy, the top one, means you spend your life living in the salt water, but you have to come back to fresh water to spawn. And that generally means the progeny, the eggs and embryos, salt water is probably lethal for it, and they have to come into fresh water to spawn. And then the fish, in salmon's case, they go through some really dramatic physiological changes, allowing a freshwater fish to move into salt water. So if anybody's a fisherman, if you take a freshwater fish and throw it in salt water, it dies. And if you do the same, you take a saltwater fish and throw it into the freshwater, it dies. So this ability to, change your physiology in order to go from freshwater to seawater it's time consuming, and everything has to happen in the right order for that to succeed. So then the opposite of that strategy was called catadromy, which means you spend your life living in the fresh water, but you go to sea to lay your eggs. And I'm sure you're familiar with the American eel, which exhibits that life history. And what's remarkable about the American eel is that all the eels on the East Coast, when they're ready to spawn, they go out into the Sargasso Sea, and they all meet there and spawn. And that's not the incredible part. When their youngsters hatch, it's called an elver, and there's big money to be made by catching eels in some of the rivers in Maine. They repeat that journey. Fish this big go from the Sargasso Sea back to its natal river. So that's I think pretty spectacular. But if you catch a bucket full of them, you can sell them to the Japanese for about $100,000. So it's a very lucrative and highly managed fishery in those northern New England rivers. Potamodromy means that the fish lives in the fresh water and it spawns in the fresh water, but it still makes a migration. So here I have a northern pike. It's catching fish in the lower part of the river and life is good. But when it spawns, it still has to make a migration up into stream waters. And the lake sturgeon is a good example for that as well. They live in the Great Lakes, but when they spawn, they go into the tributaries, and they're as vulnerable to impediments in that migration route as sea-running fish. And then finally, amphidromy is a migration between salt and fresh water that's not spawning related. All right, so when a striped bass comes up and you catch it in the fresh water, it's not there to reproduce necessarily, and bluefish do the same thing as well. So they're moving in and out of these environments, not spawning related. And actually, striped bass do spawn in kind of brackish water. Nevertheless, there are still movements that, can a fish go someplace else to find food? Yeah, probably. But these are behaviors and life history strategies that have evolved over a long time. So, if they're impeded from accessing this food resource, there may be some longer term effects. But those kind of questions are harder to answer. Just a real quick blurb on, this was a poster made for one of the American Fishery Society's meetings, and it outlines the sturgeon's reproductive cycle. And all sturgeons kind of follow this basic plan where, the adults lay eggs in fresh water. We hatch into this kind of tadpole sort of free embryo, and that develops into what actually looks like a small sturgeon, and then depending on what latitude the population is, depends on how that fish behaves. But I'll talk a little bit more about that in just a bit. So, where are we with sturgeons today? So this is a topic called global decline, and these are the four major impediments to sturgeons throughout, our coming onto the scene, especially with industrial might. So migration obstructions. Now, so this is a picture of a dam, and I know it's hard to see here, but all these little black specks here are sturgeon fins. So this is a group of sturgeons that we want to move upstream and lay our eggs, and they can't. So they're all stuck here behind this impediment. In some cases, they can manage some successful spawning, but it's not what they were evolved to do. It's not optimum success. And in many cases, if they do spawn at this site, their progeny can be too close to salt water in some instances. In fact, there's a population of sturgeons in the Carolinas where scientists believe that's exactly what happens. They come up to the first dam, which is only about 30 kilometers, 20 kilometers from the sea, and they spawn, they catch eggs and embryos, but they cannot find recruited youngsters. So it's believed that the salt water is destroying most of them. Overharvest, as I was explaining to someone else earlier tonight, sturgeon flesh it's good, it's okay, it's not ooh-la-la. But what really attracted the harvesting effort was the caviar. Caviar is worth its weight in gold. I think right now an ounce is selling for about $180. I haven't been kept. I did buy some one Christmas from a manufacturer, and I think I let it sit too long. So anyway, you're supposed to eat it right away. The best caviar, I'm told, is right out of the fish. So it doesn't age well, and they ship it in these $100 shipping boxes. Anyway, that's what really drove and continues to drive the harvest. And unfortunately, the fish has to be destroyed for those eggs to be harvested. And I'll get to that in just a bit. Habitat alteration. This is taken on the Kennebec River, and just downstream of this. Now this is an emergency spillway. The powerhouse is down here. This is actually a fish ladder. But they have this emergency spillway in case water has to be dumped so it's not flooding people's basements. But just downstream of this is a known sturgeon spawning ground. So what do you imagine that can do to delicate eggs and embryos that are just 100 meters downstream of it? And then of course water quality. This is just a random photo of a fish kill. And the good news about that is, the New England rivers and the Clean Water Act have come a long way in being cleaned up. And if you build it, they will come. Excuse me. I was talking to somebody tonight about during COVID, all the boat traffic stopped in Venice, and you could actually see the bottom of the canals, and dolphins came back in between the canals. So if we can manage, or can find ways to get out of the way in some instances, nature can recover. And so, this is my slide on the caviar fishing. This is a white sturgeon from the Columbia River Basin. This is an Atlantic sturgeon, I think from the Hudson River. So, there were active caviar fisheries here in the States. And this is the ovary. It's just a big mass of eggs. And well, why don't you just wait till the sturgeon's ready to spawn and harvest those eggs? Well, they've changed a great deal by that stage. These eggs are small and dense, and they're still. Each egg has a tendril that provides nutrients to it, and that's all still bound up in this mass. And you actually have to rub the eggs over a screen to kind of tease them apart, because you don't want stringy egg masses on your cracker. but so you cannot harvest quality caviar, because when they get ready to spawn, the egg takes on water. It gets like two times the size, and it's just no fun. So unfortunately, the caviar industry demands that this magnificent female who's done everything right and can carry a population for a decade has to be put down. So that's that story. In the United States, on our East Coast, there are currently only 14 remaining short-nosed sturgeon reproducing populations. And here's the Merrimack. So all these rivers have been demonstrated that there is successful spawning going on. Some better than others. The largest population we know of is here in the Hudson River. But you'll notice there's kind of a big hole right in here in the mid southern Atlantic states. And some of those populations are gone completely. So how do we learn all this stuff? Well, for my job, you know, I'm a field biologist, so I'm going out there and asking the fish, you know, what areas are important to you? And I don't speak their language, so we have to get creative. So here's just a millennia-old technique for catching sturgeons. This is a gill net. Humans have been using gill nets for a long time. Now, what happens if you catch a salmon or a shad in a gill net? They'll fight till they die. They don't want to be bound up. They're very uncomfortable in a gill net. These guys, they'll just kind of like, they won't fight, they won't struggle. We set nets overnight, so the fish could be in a net for 10 hours. So they're very, they have a demeanor that, you know, they don't get overstressed. They can endure problems like this. So this is the weapon of choice for catching sturgeons. Years ago, you know, and what's interesting about this line of work is, you know, every few years, new technology emerges. So you can fill in those holes, like, boy, if I only had a way to, well, if you wait long enough, that might actually happen. So right now we're in the age of, you know, microelectronics. So we can shove a tag inside of a fish, and this tag is an acoustic transmitter. So I have to put a hydrophone in the water, or either from the boat or from an anchor, and this will transmit for 10 years. So I can track an individual for, you know, a long period of time, and which makes the data about what it needs a lot richer. And for sturgeons, I think I failed to mention this, they can live, they're very long-lived fish. Shortnose are, the oldest fish is estimated to be 64 years, and there are those who think that there's white sturgeons that can reach over 100 years. So they're good at what they do. And so for a species like a sturgeon that, well, one of the big questions is, you know, how does it spawn? Where does it spawn? You know, how many times does it spawn? You know, to put a tag in there that lasts a year, you couldn't get that information. But we're looking at fish spawning periodicity, and I'm seeing I can see a fish spawn 3 times. If it spawns every 3 years, I can have this female tell me, where are you going to spawn? When are you going to spawn? So that data set becomes a lot more informative. Then, of course, we can also put tags externally. This is a smaller tag. I just need information from this tag for 6 months. So we don't put the fish under the stress of a surgical procedure. We just kind of put it through the stress of having some holes drilled through its fin. And this with the antenna is a radio transmitter. And the benefit of the radio transmitters is I can put an antenna up on my boat and cruise, and I can survey kilometers of river in a short time. If I have to put a hydrophone in the water, now I have to go at really slow speed, and I can't cover as much time, as much space. We've learned that males and females exhibit different behaviors. If you're looking for, trying to identify where spawning is occurring, don't ask the males. They're just looking for the females. I'll go anywhere you say. The females are the ones that decide where and when to spawn. So if you catch a sturgeon in the field and say, well, let's tag it, like, well, what questions do you want to ask? So, cutting each fish open to see if it has eggs is not approved. So we developed, this is a fiber optic scope. The tube that eggs travel through as they're exiting the body is translucent. So we can shove a fiber scope up there and actually look and see if there are late stage eggs in there. And this is a photograph through this device. These are, we call these early stage eggs. They're smaller, they're orange. Could be 2 or 3 years before that fish spawns. This fish, however, is late stage. The eggs are bigger, they're darker. That fish is going to tell us something in the spring. So we'll tap that. So once we have tags in a fish and they start to exhibit behavior that, hey, it looks like they're queuing up for spawning. There's both males and females coming to this spot at the right time, the right substrate, but how do we really know that they're spawning? Well, we developed this technique to try to answer that question. So we're not trying to catch the adults, we're trying to catch the progeny. So this is our sampling rig. This is basically a plankton net, and we made this kind of D-shaped opening so it's closer to the bottom where the spawning is occurring. So these are fertilized eggs, and if you look close, you can see there's actually the backbone is starting to form. So if I catch one of these, I know that spawning succeeded, at least a little bit. So one female and one male got together and they made youngsters. Now this is an example of, when I showed you that picture of the disturbance upstream of a spawning site, when these eggs first come out, as soon as they touch water, they get really sticky. So when the female says, I want to put my eggs here, they actually stay where she puts them. So then imagine this big release of water comes along and detaches some of them, and they roll along and collects sand grains. So now instead of sticking to the rocks, they're sticking to these sand grains, and they continue to roll downstream away from where the female selected. So that's, one of hydropower's practices is some cases really dramatic changes and unnatural changes in water flow. So it takes about 8 days after the eggs are laid before they hatch. And this is called a free embryo. And if you'll notice, there's no eyes, no fins, just a huge yolk sac. So what the fish does is, these embryos, they look for spaces in between the rocks. If there's no spaces in between the rocks, they can't find protection where they can live off of this yolk sac and start to develop their sensory capabilities. So if you were to spawn in sand, these eggs would keep rolling and maybe eventually hatch, and then when the fish hatches, it's got to fend for itself without being able to really sense its environment. So this rock and rubble substrate, which is characteristic of swifter waters farther upstream, is perfect for that. So the rocks are brushed clean of debris, so the eggs can stick to them, and then after the embryos hatch, there's places where they can get in underneath it. And then they start to develop eyes. You can't really see it too well here, but there's barbels here. So after about another 8 days, they start to actually look like a little sturgeon. And once they reach this state, they can swim, they can eat, they can see, and their behavior changes from let's hide under the rocks to let's swim up in the water column. And they let the natural flow of the river disperse them downstream. And that's kind of how they manage. So anything that gets in the way of that water flow, to provide the right substrate and to provide the out-migration flows can really have an impact on how successful that spawn was for that year. Okay, coastal migration. Now these are the data slides, and I did my graduate work here starting in 1987 to 1990, and we're going to talk about this stretch of the river. Here's the Essex Dam in Lawrence and River Mouth, and these are collaborators. So we came over here, and before I get too far with this, this is just some, how do you know sturgeons were here? This is a 1634 account of sturgeons, the shoals of Cape Cod and the river Merrimack, where much is taken, pickled, and brought for England. And then in 1833, there was another accounting of sturgeons jumping up and falling into people's boats. So this sturgeon jumped in and fell into the boat at the Lowell's boat shop. And then this is a Gulf of Mexico sturgeon, subspecies of the Atlantic, coming way out of the water, high enough to clear boat rails. So they're capable jumpers. And why do they jump? Scientists have looked at that, and there really isn't a definitive answer as to why they do this. One theory is that their feeding is so good that they leap out of the water and make a splash just because they feel like it. But that splash tells other sturgeons that, hey, maybe this is a good place to go. Others say that no, that they're shaking the food down in their stomachs, but so the answer is we really don't know. But they are capable jumpers. But unfortunately, unlike a salmon, they're not able to leap up over waterfalls. So this is more like seeing an otter jump than a fish actually trying to accomplish something. And if you want to see jumpy sturgeon, I know many of you have, some a little bit too closely, go to the Joppa Flats in April, May, and June. And most of the fish that you see coming, sometimes they come all the way out, sometimes they just kind of flop over. Yeah. That's a real busy time for sturgeon, because I'll tell you why in just a moment. So during my graduate work, at 4 years of effort, we set 11,000 gillnet hours. And a gillnet hour is a 100-meter gillnet, length of a football field, set for 1 hour. So that represents a lot of effort. And we caught only 24 short-nosed sturgeon in that whole effort. So we did a mark recapture estimate, and we estimated that there were 32 adults living in the river, and these are confidence intervals which say, well, it could be between 20 and 79 fish. So we say that there were less than 100 adult sturgeons in this river at the time of this study. And then we caught a few Atlantics, we identified foraging habitat, wintering habitat, and we did document some successful spawning. So steam ahead. So after that was done, I started working at the Conti lab, and I hadn't returned to the Merrimack till 2008. At that time, there was interest in gathering tissue samples from all the short-nosed sturgeon species throughout the entire range, which is Georgia to Canada. So they could do a comparative estimate, evaluation of the genetics, and there are other things we can learn about their distribution and movements and population dynamics through genetics. And that's the case of the technology advancing and providing new opportunities. So I was asked to go back to the Merrimack, and Micah, could you, if you can catch 5 fish, that would really be helpful for us. So back I went, and over a course of 8 years, we set only 1,800 hours of gill nets, and we caught 505 individuals. And we estimated that there were now almost 2,500 adults in the river. So something remarkable had happened. And I was all set to, I'm going to be able to write new chapters about, these were the first females I ever captured. So I was all set to see my name in lights. But what we found was that 80% of the females that we tagged that were full of eggs left the Merrimack and went up and spawned in the Kennebec River. There were 3 known identified spawning sites in the Kennebec River. So this is the Androscoggin River, this is near Augusta, and this is near Waterville. But I have the Edwards Dam here highlighted. The Edwards Dam was removed in 1999. So this spawning site had been cut off from shortnose sturgeon until 1999. And what was amazing to me was that the very next spring, after this dam was removed, there were sturgeons up here in Waterville spawning. And some of those were fish that were tagged in the Merrimack. So we learned at that point that there's some real interesting dynamics going on between the Kennebec and the Merrimack. And in fact, I don't have it pictured, but the Penobscot River is farther upstream here, and the same thing was happening. There were fish that were living in the Penobscot River, but they came to the Kennebec River to spawn. The difference was that there was no spawning in the Penobscot, but there still was spawning in the Merrimack. So if you've read about the last glaciation, the glaciers covered New England all the way down to Long Island Sound. So all of these New England sturgeon populations had to have been recolonized, and they are a colonizer species. So we were wondering, are we actually seeing colonization going on? Is there interbasin spawning, or fish spawning in the Merrimack some years and spawning in the Kennebec other years? So that study is ongoing. And as I said, we can put these 10-year transmitters in a female who usually spawns about every 3 years. So we're slowly collecting information on how much interaction is there. Genetically, they're very similar, but behaviorally, we're still trying to work that out. But the last time the data was reviewed, we had 4 individual females that had multiple spawning years that we witnessed, and if they went to the Kennebec one year, they went to the Kennebec all those years. So I refer to those fish as the Kennebec marauders, because they're coming down here and competing with the Merrimack population for the resources, the foraging. And is that a problem for this? This population may still be hanging on by its fingernails, and everybody else that has come to the river is there because they can't make a go of this. There's too many fish in the Kennebec now for everybody to be happy and find the resources. So they left. Some go to the Merrimack, some go to the Penobscot. So that's an interesting little bit. So moving on from that, they also do something rather peculiar in the winter. So winter boating is as fun as it looks. The traffic is great. But here is a photograph of a camera hung over the side, and this is what sturgeon do in the winter, at least at these latitudes. When it gets below about 7 degrees, they form these aggregations, they stop feeding, and they just sit there for 5 months. And there was a professional hydrographic survey team that came in with a side scan sonar and took these images. Now, this part is actually the sonar shadow, but you can definitely see it as an outline of a sturgeon. The actual sturgeon is this white patch right here. But nevertheless, again, this rise in technology, and this is used by numerous sturgeon investigators. If the fish are still, you can get these spectacular images. And this is right off of Hales Island, for those of you who are familiar, just below Haverhill. And this is where they're spending the winter. And the power with this technique is you don't have to do mark recapture estimate. You can just count them. And they came up with 3,786 individuals. So that's, maybe the population's grown a bit, maybe I missed a bunch, you know, when I did my estimate. Because market recapture, I did that in the winter. So not only is it delightful to be boating in the winter, but then to be hauling nets and working with fish. So this is a fantastic technique to throw at this kind of thing. So last thing I wanted to kind of cover was, so sturgeons are living in this remarkable community of organisms and a dynamic river system. So what? So I was a participant in a foraging study of shortnose sturgeon several years ago, and the major prey species that these fish are seeking out are sandlance. I don't know if you're familiar with sandlance, but they're in such vast schools that whales feed on them. But when they're youngsters, they come into the mouth of the Merrimack, and I witnessed them in the shallow water just dive into the sand. You know, they swim through that hydrated sand, and that's part of their defense mechanism.
Exactly what we call a sand eel, right?
I think that's the same thing, right? And they get to be, well, that's a big one, I guess. But when they get larger, they move offshore and to a place where whales can exploit them. The second real big prey item are these amphipods, and they're sand fleas. I'm sure you've all been disgusted by them, but they made up about 95% of the prey species in the short-nosed sturgeons that we sampled. And the sandlance made up about 90% of samples in Atlantic sturgeon. So there's bivalves, you know, mussels, snails, clams, anything that's small enough for them to swallow. And they have if you dissect them, they have it's almost like a bird's gizzard. It's a muscular organ that kind of helps break shells apart. So they eat shells and they eat pebbles, and then they kind of mash it together to help break some of the shells apart. But I've captured sturgeons in the Kennebec River and brought them back to the lab for us to study later. And the next day I walked in and there's, what's all this stuff in the tank? And here there were hundreds of these pinhead-sized snails climbing up the side of the tank. So they didn't get to eat all of them, but they were also maybe able to transport living organisms around. And that's not necessarily a bad thing until a few slides from now. And then, of course, polychaete worms. I pulled my anchor up from the Joppa Flats one year, and there were 30 polychaete worms in the mud on my anchor. I was like, that's why they're here. And that was not correct. Probably takes a lot of work to dig those polychaetes out of the substrate. Their tummies were full of these amphibians. Now I bring up invasive species. And here's the same picture you saw yesterday, or last slide. This is the Asian clam, also called the Asiatic clam or corbicula, and it's came over probably in the ballast tanks of cargo ships. But I was working on the Merrimack some years ago, and these appeared in the fish holding tank. I'm like, well, I know where you came from. So I made a note of it and didn't think more about it. And later realizing I should have really determined if they were living or not. Some of them were crunched up, some of them were not. But just a few years ago, the same thing happened in the Kennebec River, and this time I thought, I'm gonna find out. And I held one in my hand, let the oxygen deplete, and it opened up. So it was alive. So sturgeons are able to transport an invasive mussel like this around and redistribute it. And I don't know, I'm sure you've heard about the zebra mussel. Not so much a problem here at this point, but definitely in Lake Champlain. And the theory was, well, let's build the sturgeon population up because they're going to eat them. Well, they're going to eat some of them. So the same thing's probably happening with the zebra mussel and lake sturgeons in that their cruncher doesn't get all of them, so they're probably helping this invasive species to move along. I don't know if you've heard of the green crabs. We haven't seen any evidence that sturgeons are feeding on green crabs, and why do we care about green crabs? Well, first of all, they're doing rather well, and when a species comes in and it does establish itself, it can kind of rewrite the ecology of an area, which may impact the food that sturgeons are eating. Now, we don't really know the answer to that. I'm not a community ecologist, so I can't say much more about that. But the one thing that can happen is take the cod, for example. We as humans have harvested a lot of cod, and there's measures to be put into place to try and let the populations rebuild. But when you remove a significant part of a population from the ecosystem, from its niche, something else can kind of take its place. And I think with the cod, the spiny dogfish have come in and say, well, if there's nobody here, we'll just move in. So it's not necessarily always a matter of, well, let's just cut back on fishing pressure and the stocks will rebuild. Someone may have already moved into that house. So, in order for the cod stocks to rebuild, those dogfish have to get out of there, and they're very adaptable and resilient species. So, when you have shifts in ecology that could be brought on by invasive species like this, even if you were to remove all the green crabs, there may still be something that gets in there before the native species and reoccupies that resource. And then, I don't know if you guys, do you guys know there's water chestnuts in the Merrimack, yes? Okay, so we have, I think we have a bit more of an infestation on the Connecticut. So the Kennebec River Watershed Council has chestnut pulling. Do you guys do that too?
The
Okay.
Mystic, they do it on the Mystic.
All right. And then I know this is a much discussed topic here, as probably many rivers that combine sewer overflows. This is just a stock picture. I don't know if it's an overflow, but it's the combination of sewer and storm runoff. It's untreated discharge, and I'm not the expert in the room. I think John knows a whole lot more about the details of this, but they're a safety precaution if they don't, in many cases, if they don't allow this untreated material to enter the river, it can come back into your homes and flood areas. So it's a safety practice that I know it's being, alternatives are being worked on, but what this can result in is excessive algae and bacterial blooms, which, the algae and the bacteria are always out there, but when they get this massive dump of nutrients, the populations can explode, and that can result in oxygen depletions. And then when these blooms run their course and those organisms die off, they sink to the bottom, and they can still consume oxygen and create dead zones. In fact, there was a sturgeon dead zone in the Delaware River near Philadelphia, because there was so much oxygen depletion occurring because of the practices of the city, that there were sturgeons above and below, but they didn't hang out in this oxygen depleted zone. And then, of course, the loss of ecological niches that can take long times to establish. And when our activities go in there and stir things around, it's not always necessarily just, well, let's clean it up and it'll all go back to the way it was. Some cases that can happen, but there can be much longer lasting effects. And finally, I'll talk about pharmaceuticals, which I've got human waste, agricultural waste, and the pharmaceutical industry effluents, are all contributors to pharmaceuticals getting into the waterways. The most common compounds are, anti-inflammatories, pain medications, and this one, the antibiotics. You may think, well, how often do I put an antibiotic? It's not that it shouldn't be a problem. But the issue really is with the use of antibiotics in the, livestock. So they're pumping massive amounts of antibiotics into livestock that gets passed out and winds its way into the river. And endocrine disruptors, pesticides and hormones, this kind of compound can actually impede reproduction in fish. And there were cases in the Potomac where fish were growing both sex organs in the same fish. So, and I don't think that it's fully understood exactly what these effects are, but that's one of them. Aside from the antibiotic load, the endocrine disruptors have, at least for fish, have people relatively concerned about perpetuation of established populations. Is it enough three-syllable words in a row for you? Okay, and I'd like to, I kind of take pride in the ability to, as I said, when I'm out in the field and I have a sturgeon on board and I see somebody close by, I'll go to lengths to try and, hey, do you want to see a sturgeon? And I tried that at midnight, and they weren't having it. But especially when kids are on the scene, this was these are the Lowell's Boat Shop boats, and they had a class out in the dories, and I got a call about this big dead sturgeon. I'm on the sturgeon carcass recovery team, so I talked to Graham McKay, and we went and picked this thing up near Newburyport, and he said, if you have an hour, I'd really like to take this fish up to my class. And so we motored up in a homemade boat, and he was always clucking about, I don't know if this thing's going to hold water. But it was a boat that his students made, so we lumbered up there, and we spilled out onto the sand flat, and I gave a little lecture about this sturgeon for his class. I didn't come up with it. Someone else did that. But I thought it was a good idea. And this photo was taken in Amesbury. Well, that's what I got for you guys. And I am able to respond to questions if, I don't know, okay.
I take it the sturgeon provide no food value. You have any comments on that?
Ask a seal. But Native Americans survived on sturgeons. After harvesting the caviar, those carcasses the meat would be smoked or pickled and shipped to Boston. So yeah, it was a staple food source, particularly during the spawning runs when there were a lot of fish predictably moving up into the river. I read an old story that said it was dangerous to be in a canoe during the Atlantic sturgeon spawning run because 12 to 14 foot fish with raging hormones. So you could be in a tough spot in a boat during that time. Or if you're down in Joppa Flats. Yes?
Where does the Merrimack kind of go from salt to brackish to freshwater?
We measured that back in the 1980s. And so we measure, salt water is heavier.
Yeah.
So when it comes up, if you really want to put a nail in that, you have to put your salinity meter down on the bottom. And it was the big wide part of the river in Amesbury, goes in those nice scalloped edges. Right when that starts to constrict again upstream, that's about as far upstream as salinity penetrated.
Okay.
And it wasn't seawater at that point, it was like 5 or 4 parts per 1,000 of salinity. So just barely getting up that far. But one of my jobs is to revisit that, because I've noticed things have changed, and the location where the river actually reverses direction has gone up, is now farther upstream than 20 years ago. So I know that the salt wedge has got to have gone up farther, and that's something we're going to measure, in the next few years.
That varies a lot by the weather.
Indeed
This
it
year, I'm sure it penetrates much further up than last year. It probably didn't get beyond the north end boat club, to be honest.
Right, and
So
it
it varies tremendously.
depends on tide, it depends on rainfall, depends.
Rainfall, yeah.
So when we take those measurements, we wait for the highest tides and the lowest river flows. So we let the salt penetrate as far upstream as it can, just to give us that boundary. But you're absolutely right, that is a dynamic feature.
What is it about that part of the river out near Haverhill that makes it such an attractive place for them to overwinter? I mean, why would there be nearly 4,000 sturgeons just hanging out right there?
If you can answer that, you get a raise.
I mean, is it something about the bottom, or the water clean, or what?
We've looked. I mean, we've gone to a winter site in the Kennebec River, and we've measured. We did a grid. We measured temperature, salinity, substrate, discharge, or dissolved oxygen, and we couldn't find anything different about where they hung out compared to 5 and 6 kilometers in either direction. Like, guys, what is it about this spot? We thought maybe that there may be some freshwater springs that are entering the river at some point, but we never were able to put our finger on that. And in addition to that, sometimes these migrations meander. When I did the population estimate, the mark recapture effort, the winter sites were in Amesbury, in that real wide part of the river. And now there's hardly any that spend the winter there, and they're spending winter farther upstream. I'm like, is it because of this? Is the salt coming up and disrupting that? I don't know the answer to that, but that's something I'm thinking about.
But they can't get beyond that dam and line.
Not without assistance. I don't think they've ever lifted a fish in the elevator there. But I will share this with you. I received a videotape or a cell phone recording from somebody that I cannot say that it was not a sturgeon. And this was upstream of the Amoskeag Falls. And so, historically, they inhabited the river at least all the way up to the Amoskeag Falls. So are there still remnant bits of the population between and upstream of the dams? We haven't really looked.
How stable is the population?
When I did my mark recapture estimate, I got about 2,500 fish. When the consultants did the side scan sonar, they got about 3,500 fish. Now, you could say that that's showing an increasing trend, but there's always a lot of uncertainty around those estimates. So in my mind, they're similar. And if I'm around for another 20 years, maybe I'll have a look again. But it could be indicating a trend in more fish. But I would call it stable at around 3,500 adults. Yeah.
I'm curious about the evolution of this fish. Because I've always heard it was an ancient fish that was not seen for a long time and now it's seen. But the hard shell, that it's still the same as it was all those thousands of years ago. Is there any theory as to why it has not evolved into a softer shell? Is one question. The second is, who is its predator? Who eats this thing?
I've seen, scientists have seen seals come up out of the water with a sturgeon in its jaws. I've seen an eagle. I have a picture of an eagle trying to pull a sturgeon out of a stranding pool. But those are just the adults. Maybe sharks go after them. I don't know that. But the youngsters that can be swallowed whole, anybody with a big enough mouth, I think would be preying on those as well. And then, of course, we're in that mix to some degree, but, and I've also, sometimes the tag stops moving, so either the fish is tired or something's happened to it. And we've learned that they can eject the tags, but we also know that they can die, and I had my little antenna receiver, and I'm like, the tag's up on that hill? So I climbed up there, and lo and behold, there was the tag, and it was all kind of crunched. So I think the fish died, and then a coyote or something got hold of it and dragged it up the hill and chewed on it. But I don't know that.
And the evolution, how come it has not
Well,
changed its form?
Probably because it hasn't had to. The ones that evolved the armor, survived to reproduce another day. But there's nothing that's the presence of that armor isn't impeding it in its survival and thriving. So we don't have to. I'm trying to think of an example, but why does an elephant still have a trunk? But, well, that's obvious. So it can trunk it. So I mean, I don't know. I'm not an evolutionary biologist, so I'm sure somebody would have an idea or a thought anyway. Question?
What's giving you the greatest joy out of this adventure you've been on to become one of the world's experts?
This scar right here. There's one other person in the room with a sturgeon scar, and she beats mine. But no, just when I'm asked that question, like how do you get here, and my simple answer is I kept following the path of least resistance. It was animals and water seemed to be my guiding. If I had a choice between, do you want to work at, you want to sell fire extinguishers and retire early, or do you want to go on a Japanese fishing trawler? Well, I like sushi. so that I'll add this. When we were crafting our radio tags to fit inside the sturgeon, they have this really long antenna, and I had to make it shorter. So if I just cut it, it drastically reduced the range. So I actually wound it. I made a coil, and I potted that in this material that they use to coat pacemakers. So it's kind of goo, it hardens, and it's nothing can attach to it. So that's what we were using. And I had to use needle and thread to bind it all together. And I was thinking, when I was younger, I had a creepy crawler set. And I used to make these little rubber skeletons. And then I'd throw them into the lawnmower and sew them back together. I know how to do this. So yeah, I don't know that I could have ended up anywhere else. Yeah.
You mentioned that the spawning happens every 3 years, but it sounded like the fertilized eggs are in the female just for a short time. So why is there such a huge gap between one set of eggs and the next?
That was a generalization. A lot can affect how well a sturgeon can reproduce a batch of eggs. For a female, that's a lot of work. There's a lot of energy, the yolks and all that. So it can take, and it differs in different rivers. If you've got really cold climate that you're living in, it probably takes a little longer. If you're living in a very hot climate, it may take a little longer. If you have a really bad fall foraging, the fish are out there now feeding, preparing for the winter. So this is a really good time to study sturgeon feeding. And if there's bad weather and you fail at putting on all that fat and resource for the winter, you may not have enough energy left to make eggs. Males can males can just they usually come up to the spawning site every year, because sperms are a whole lot cheaper to make compared to eggs. So it can depend. A virgin female may take, longer to produce her second batch of eggs, where these large successful females may be able to turn that around a bit quicker. We've actually had a case on the Kennebec River where it was an unusually warm winter. So we think when the water temperature gets cold enough, their metabolism slows down. So they don't need to feed, they don't need to spend as much energy. But during this warm winter, it probably was a little bit more energetically costly just to exist. And after that spring of that warm year, we didn't catch any sturgeons on the spawning site. So it appeared as though it was a very energetically costly winter, and it just tanked the whole spawning year. Yes?
This is a comment. There's a lot of people here that have seen sturgeon jump. There's probably a lot of people here that haven't seen sturgeon jump. And I was just gonna describe, if you're in doubt, that sturgeon, usually they don't, the tail doesn't usually come out of the water.
Right.
They usually come up like this, and they go right straight down. It makes a loud noise and a huge splash, like a belly flop.
Right.
And no other fish does that I know of.
Right. Just for on your way out, I have some posters that kind of reflect some of what I've talked about and maybe a little bit more. But on the table below one of them is a bunch of sturgeon sputes that you can look at and play and wonder how this would feel raking across your face. But most of you. Okay. Thank you.
Micah, thanks very much.